Lab number 11, embryology sponges as well as the Cnidarians. So the jellies, we have the stinging jellies and the comb jellies. So here we go. Anyway, showing the process of gastrulation, and once again, remember we have zygotes start dividing, creating a hollow ball of cells known as the blastula, and that particular blastula will start collapsing. In this particular case, it start collapsing into the blastocell. Remember, cell is soil is basically the cavity. So whenever we start talking about that, we are creating from the blastula, it goes into a next step known as a gastrula. So now we're looking at the ectoderm, endoderm, and then some of the cells will start going in between, creating a mesoderm. So each one of those embryonic layers will give a very particular systems of organs, right? We discussed that. So let's see, let's move. Here we're looking at the overall table. Make sure that you understand that, because you will need to kind of flip it over, not literally flipping over. Don't do something like this. I had students doing something like that before. No, I need something similar to that, but starting with animal multicellular on the very bottom part of next lab, I believe. Yes, not this lab, next lab. So anyway, pre-lab questions. Is the zygote haploid or diploid? The zygote is a merge of haploid egg and a haploid sperm. So haploid 1 + 1 will give you 2. Gotta be diploid. What is the name of the hollow ball of cells? The full ball of cells, known as the morula, we'll talk about that today. The hollow ball of cells, known as the blastula. What is the stage when the ball of cells starts to fold inward? And so as soon as we start talking about folding inward, collapsing on its own, we start talking about the gastrula. Or what is the stage when the ball of cells start fold inwards and create the arterotron? It's basically the ancient gut. This is what will turn into the digestive system, and that's a gastrula again. Name 3 germ layers in a gastrula. Ecto, outside layer, ectoderm, inside layer, endoderm, and in between layer, mesoderm. So ecto, endo, mesoderms. How many germ layers bilateral? Bilateral have 3 germ layers, ecto, endo, and mesoderm. The radiata, such as jellyfish, will have only 2, ecto and endo, not much of the mesoderm. Are animals eukaryotic or prokaryotic? Is the animal a bacteria? No. Are you a bacteria? No. If you are not, then you are eukaryotic. You have nucleus, membrane-bound organelles, mitochondria, Golgi apparatus, and so on. Define heterotrophs. Heterotrophs must consume organic compounds. They cannot make their own organic compounds, unlike plants. And that was one of the major things for the plant. Remember, there was a question regarding the role of the plants in the biosphere. Well, many of you says oxygen. I agree with you. Oxygen is important, but the main thing, they are producers. What do they produce? They taking carbon dioxide and they producing organic compounds, sugars. Well, so later on we can enjoy those ones, right? What are the invertebrates? Invertebrates are a group of animals, a very large, the largest groups of animals, which does not have a vertebral column. Which supergroup does the kingdom Animalia belong to? The same group as the choanoflagellates and the same group as the fungi, known as Apistoconta. I think there should be the name. Yeah, here we go. Apistoconta right here, supergroup. Pista conta. What are the three characteristics used to classify animals? Well, eukaryotic, right? Let's see. All animals, multicellular, heterotrophic, eukaryotes. Same as these fungi, if you think about it, most of the fungi are multicellular, they are heterotrophic and eukaryotes. That's one of the reasons why we belong to the same group. All right, table number one. In table number one, you have just the pictures. So you have a pictures of like a jellyfish, and then you will have or any kind of, well, that's a coral. I took that particular picture. In the actual Mesoamerican coral reef, I think. Here we have some more, and that's not the coral. That's actually a crinoid. So in this particular case, we're looking at the radial symmetrical creatures, right? Radial symmetry. So symmetry radial, germ layers 2, ectoendoderm. That one is, I probably need to move it away because that one is a relative of, that's the deuterostome. That's actually triple 3-layered. But yeah, generally, if you see a jellyfish or coral or anything like that, there will be a symmetry, radial symmetry, and 2 embryonic layers, ectoendoderm. For the bilateral symmetrical, here we go, bilateral symmetry, left side very similar to a right side. Here we go, and that's the gray monitor right there. So those ones, bilateral symmetry, right? And the germ layers will be 3. Which ones? Ecto, endo, and mesoderm. Yeah, here we have the beautiful picture. Literally kind of in my backyard, took that one. That one is, again, bilateral creature, right? And that's a Texas paintbrush. It's in Texas, near Austin. So anyway, so all animals can be subdivided into 2 large groups, right? No true tissues, parazoa, and true animals, meta or metazoa. And here we're looking at the radial versus bilateral, Cnidarians and Ctenophora, and we'll talk about that today. So, but before we'll go, protostomes, deuterostomes. So embryonic development, define the term below and label which picture shows the term. Okay, so in this particular case, let's see the protostomes. Protostomes, what exactly protostomes will have? This particular invagination, the first part, right? In this particular case, whenever the gastrula start collapsing, that opening, the blastopore become mouth. All right, that's the protostomes. In deuterostomes, the blastopore will become anus. All right, drawing the body cavities. Drawing the body cavities, you basically can pull out the pictures from the lecture, or you can literally draw. It's not very hard. You can draw. Let's see. Here is the ectoderm, right? Then let me use the. Let's go with the yellow. Endoderm, and then let's. Well, actually, usually the ectoderm goes outside, so no body cavities. So here we're looking about the ectoderm, endoderm, mesoderm. So ecto. Whoops. Ecto, meso, and so in this particular case, the main idea that there is no body cavity. So all of that is filled up with something. Now, in a pseudo-coelomates, you will have exactly the same picture as well as in the coelomates, exactly the same thing. But in this particular case, let's go with the red one. So we're looking at the mesoderm. And then we're going with the endoderm, right? The digestive one. So the beginning is pretty much the same, almost. So that would be endo. And then that one would be a meso. Sorry, not the best. What's the difference? So here we're looking at the, what's the difference between all of those? Here I need to outline right here. Okay, so now look, got it. Here, all of that is filled up with a gel-like material, so there is no cavity at all. No cavity. Now, let's take a look on the cavity. Here's the cavity located between the endo and mesoderm. That's a false cavity. And now, cavity within the mesoderm. Make sense? Yes, beautiful pictures, I know. But if I were able to do that within literally 5 minutes, you probably will be able to do that a little bit better. You will have, well, again, you can just copy and paste it, but I would recommend you to draw it. That way you can visually see it. No cavity because it's filled up. Here, the cavity, that part right here, the white one, which is located between the endo and mesoderm, false cavity. And here we have the cavity which is within the mesoderm. That's simplified speaking. I'm not going too deep into that. All right, so let's kind of move on. Embryonic development, table 2. How the zygote is formed. Zygote is formed by fusion of what? Gametes, eggs, egg and sperm. Define the term blastomere. Some of the students not really sure what it is. So here we have, let's kind of look at the word. Blasto means blastula, right? Something part of the blastula. Now mere. Mere means unit, unit of the blastula. What's the blastula is made out of? Tiny little bitty cells. Let's see, here we go. So each cell within, see the cavity, the hollow ball of cells, so blastula. Each cell right here is the blastomere. In Bio 1, you're supposed to talk about the polymers, right? Poly means many, mer means unit, many units like proteins, many amino acids combined together, right? So in this particular case, that's a kind of same idea. We're talking about the structure. Blastula is made out of a bunch of cells, each of the cell known as a blastomere. So here we go. Continue to have a blastula, hollow ball. Each one of those cells is the blastomere. Any questions regarding that? All right. Are the blastomeres haploid or diploid? Now, please remember, for the animals, the haploid structure sperm and the egg. Together, they're known as a gamete. Is it a gamete, a blastomere? No, it's not. Is it a sperm? No, it's not. Is it an egg? No, it's not. It got to be diploid. Starting with the zygote, it's already a diploid. Diploid goes through the mitosis, creating more 2 diploid cells, 4 diploid cells, 8 diploid cells, 16 diploid cells, 32, 64, 128, 256, and so on and so forth. Okay, but all of them are diploid because that's the beginning of the embryo, that's the beginning of the any kind of animal. We started like that. About first 30 minutes of your entire life, you started as the zygote. And now look at you, a couple of trillions of cells later, nice looking diploid organism, walking, sitting around, right? Walking around, sitting around, etc. Any questions, any problems on that? Hopefully not. Moving on to the next one. If giving two models, morula and a blastula, how could you decide which was without seeing the blastocell? Blastocell is that particular cavity. You count the cells. So literally 1, 2, 3, 4, it says 16 cells. Somewhere between 8 when it's a small ball, 16 sometimes to 32, but 16 is a safe bet. The ball of cells is not hollow, it does not have the cavity. It's just a ball of cells. They continue to divide, they become smaller. As a result, there will be a cavity forming inside, somewhere between 32 to 64 cell stage. So this is when morula, the stage, turning into a embryonic stage, turning into a blastula stage. That's the embryonic development. Fascinating topic. If you'll ended up going into a biology, I would really recommend you to take it. It's an advanced class, but I would really recommend you to take comparative embryology. It's fascinating how that particular thing happening, but unfortunately, we don't really have much time to spend on that. So, two models, you counting the number of cells. Morula will have about 16, give or take, 8 to 16. Blastula usually over 32. 64, 128, etc. All right. Let's see. All right, now let's label the picture of Gastrula. So let me erase my beautiful pictures right here. Markings. So Archeron right here. And some of you do that. I'm okay, I guess, with that, all that particular stuff, the inner part. But some of you do straight lines, which are much easier to see, because sometimes people do something like this, and then it's kind of hard for me to figure it out where exactly that line is pointing, or at least use a different colors. Okay, I had students before done something like that. For example, the Archeron, and then it goes right there, all that. And then blastopore. Well, for the blastopore, let's say, well, let's go. So blastopore, and then the student go and showing that that initial opening is the blastopore, right? And then the germ layers, ecto and the mesoderm. So you'll use a different color, and then we decide that I think endoderm was orange, right? So, and here it is. You can do that, yes. It's not very pretty per se, but as long as I can understand, and it's not like a web, which it's kind of hard to trace. But most of you doing a really nice job just going with the straight lines and markings there. So far, I don't really have much of the problems with you guys labeling all that. And the ectoderm, I think the ectoderm was kind of bluish. So ectoderm, the outside layer, and those pink ones are the mesoderms. All right, so now lining of the digestive tract, that would be endoderm. Okay, so here we have the endoderm. Now integument, some people don't know the word integument. Integumentary system is the largest organ in our body. It's skin. Skin will be produced by ectoderm. Nervous system, ectoderm. So we have digestive tract and ectoderm outside, right? Yeah, circulatory, skeletal system, and the muscle predominantly will be between the ecta and the endoderm as a digestive tube, and the ectoderm as the skin. So if it's in between, it would be a mesoderm. So digestive tract, endo, those two integumentary nervous system would be ecto, and three of those are meso. So we're looking at the blastula, and here we're looking at the actual model. So the blue right here layer is the ectoderm, the yellow layer is endoderm, and those reddish pink in between the blue and yellow are mesoderm. So here we go, the labels, and gives you that particular table with the correct answers. All right, yeah, here we go, the blastula, the cavities. Make sure guys that you will be able to identify the examples and what is asalamate, pseudosalamates. It will be on the test. All right, now we're moving to the sponges. Table 3, and there is a bunch of sponges we have. This is something which is known as a glass sponge, which is made out of predominantly just the silica, right? Quite often the glass sponge is a Venus basket. It will grow around the small fish, so the small fish will kind of get trapped inside. So what it actually does, the small fish will be protected by the glass sponge itself, but it's also will be eating all kinds of small plankton going through those particular openings relatively easy. It will defecate, and that particular bacteria as well as more simple protists will be attracted to that, and that would be feeding the sponge. So kind of mutualistic relationship, even though we're talking about the caged environment. So label the ostium, osculum, and spongocell. So in this case, whenever you're looking at the sponge, you see the sponge known as a perforated sac has a tons of tiny little bitty holes. Let me find this particular. Wait. Here we go. So tons of tiny little bitty holes. Those particular holes are known as the ostia. Ostium is one, bunch of them ostium. Well, bunch of them ostia, one is ostium. All right. Now, osculum, that particular part right here, right there, the opening of the where the water comes out. That's the osculum. And if I'll cross-sect in the tube, there will be the, let's see, here we go. This is those particular openings are osculum. This is one sponge. This is another sponge. Third, that's a colonial sponge. So now, if you will, in a face-to-face class, I'm taking like a pen or pencil, and I'm literally dropping it inside. And now I'm saying, now the pen or pencil is in the sponge cavity inside of the sponge. That's a spongocell. All right, so all of that deep inside there. Oh, so here we go. That's the picture. So you're looking at the top part, the opening is the osculum, and that particular black part right there, the cavity itself, is the spongocell. All right. Here we're looking at the Venus basket, also known as a glass sponge, because it's made out of silica, very intricate. You can feel that it's made out of fibers of silica dioxide, SiO2. So in this particular case, glass sponge versus, that's a calcarea. This is a bath sponge, only spongine. A little bit of calcium specules, not too much though. So in this particular case, this is what makes it soft, and that's why we can use it. It's a natural bath sponge. So now glass spicules, we'll see those ones momentarily. Let's see, I had the picture. So here we're looking at the glass spicules, 3-ray glass spicules, okay? 100 magnification. This is the 100 magnification. That's the picture of that. So Venus flower basket has only spicules, and spicules will be made out of silica dioxide. Bath sponge will have a protein known as the bath spongin, and the colonial sponge, that one will contain both of those. The spicules made out of predominantly calcium. As a result, it's kind of hard. You don't really want to use that one for the bath washing your body, because it will start literally scraping your skin off. So now we're looking at the, let's see, where is it? So we're looking at the calcarea, it's basically a lump of calcium, calcium carbonate. But when you're looking under the microscope, you will see that those particular spicules are located in a 6-ray pattern. So this is the calcium carbonate spicules, the stars kind of looking like structures. So here we're looking at a very nice. So they are specifically arranged, and the sponge creating the body around. So make sure that you will see that. It's pretty much the same picture. All right. So what type of the material makes up the sponge and the skeleton? Two, either calcium carbonate, like you see it right here, or silica dioxide, SiO2, the most common chemical on the face of the earth, right? The most common mineral on the face of the earth. So what type of the body symmetry does a sponge have? A huge letters. No symmetry. Please make sure that you understand that. Some of those particular sponges kind of look symmetrical, but technically they're not. So don't get too excited when you start seeing like, oh yeah, it definitely looks like a radial symmetry. No, it's not. It is not radial symmetry, even though once again, kind of start getting into the symmetrical shape. But sponges considered to be asymmetrical. No symmetry. Based on whatever the environment it grows in. In this particular case, let me kind of give the example. For example, if we'll put that particular sponge in the area with the very strong stream, this going this way, it will start bending that way. If the stream will change, it will start bending that way. If we look at the fish and we'll put it in the same environment, it will continue to grow the way how the fish grows. I'm simplifying it, of course, but in this particular case, asymmetrical corals, they will go in the symmetry, but it will be a radial symmetry. So sponges are asymmetrical. Please remember that. It's a commonly missed questions on the test and the lab practicals from the previous semesters. Sponges belong to a group called Parazoa. What is distinct characteristics? No true tissues. The tissues can, well, the cells within a tissue can exchange fairly easily. What type of digestive system sponge have? It does not have a digestive system. It has something which is known as a intracellular digestion. They don't have a cavity where the digestion occurs. We do. We are consuming the food that goes into a specific cavity in the tube, right? Digestive tube. This is where most of the digestive occurs. Do we still have intracellular cavity, the digestion within the cell? Yes. For example, white blood cell comes around finding the bacteria, will hug it, create the vacuole, food vacuole, will inject the digestive juices, and it will digest the bacteria. This is how the white blood cells, simplified speaking, works. They detect the invader, grab it, and digest, but that's not the majority of our digestion. Our majority of the digestion comes through the digestive system, extracellular digestion. They have only intracellular digestion. The cells grab the food, digest, spread the nutrients next to the cells nearby. Make sense? All right, now next group, Cnidarians. Yeah. And that particular picture, make sure you understand what it shows, because most likely it will be on the lab practical. We're looking at the hydra overall, that's a polyp body. We're looking at the tentacles. We're looking at the gastrovascular cavity. Now it has both. Whenever the food gets into a gastrovascular cavity, it will have extracellular digestion because digestion happens in the cavity. And then partially it will have an intracellular digestion. We'll continue to have intracellular digestion for the rest of the animals. Now here you're looking at the ectoderm, you're looking at the mesoderm. In between there will be mesoglea with embedded nerve cells. And the nerve cells will create a network. There is no centralized brain or anything like that. And we already talked about the, let's see, yeah. Define the term diploblastic. Two embryonic layers, ecto and endoderm. That's the cnidocyte, and that's the nematocyst. Okay, so the capsule itself. This is the capsule kind of springed in, and that's the release. So delivering the toxins. In the video, actually, they will be talking about the one which you're supposed to watch. On the Cnidarians, they will be showing how two anemones would be fighting each other, and they will be releasing the value of those particular cnidocytes trying to poison each other. And one of them will prevail. So define term diploblastic already did. Two embryonic layers, ecto and endoderm. Refer to the lab guide and draw the Cnidarian body plan form. You can take that particular picture of that. That's fine. Print screen it or snip it and put it in there. Refer to the lab guide and draw the Medusa body form. So here we already have how it's different. The first of all, the location or orientation of the tentacles. Tentacles up, most likely a polyp. Tentacles down, most likely a Medusa. And polyps are usually attached to the place. They're sitting, they're waving their tentacles, everything is cool. The Medusas are usually free floaters, right? That's the reason why Medusa stage is something which is known as a sexual stage, because Medusa and another Medusa will be able easier to meet and exchange any kind of genes. All right. So here we're talking about gastrovascular cavity, the bud. Here we're looking at the gonads, and here we're looking at the ectoderm, endoderm, and nerve net. So yeah. Mouth, tentacles, gastrovascular cavity. So mouth, tentacles, gastrovascular cavity, cnidocyte. Cnidocyte is right there. Nematocyst is part of it. Nerve net, you can kind of see it right here, white, tiny little bit yellow ones. And then asexual bud is right here. That's a gonad. That's another gonad. That's probably a female. That's probably male gonads. But you don't have to worry about that. What is the function of the nematocyst? Actually, two. To hunt, to kill the prey, and to fend off any enemies. So protection and the attack and protect. Hydra slide. Let's see. Here's the slide, which you will see in. So here's a hydra. Here's another hydra. You can clearly see tentacles, and on that one you can see a bud, asexual bud. That's the jellyfish, actually from. Well, it's a flipped over. So, but yeah, that's a jellyfish. That's another jellyfish. This is, yeah, Aurelia. This is the most common jellyfish in any kind of fish tanks or in the museums, etc. If you ever went to the aquarium restaurants, they have the Aurelius in one of the fish tanks, and they are glowing, kind of bluish glow when you are in ultraviolet. So you are shining the black light at them, and they start glowing. Kind of neat creatures. So yeah, here we go. This is exactly what we're talking about. Now, sea anemone looks like well, that's a preserved specimen, and this is the attachment. This is the body, and this is the tentacles. This is actually a mouth. Sea anemones, in most of the cases, they are standing, they are attached to the substrate, they are waving the tentacles. Some of them that, for example, this guy can detach itself and slowly swim away if there is any predator or anything like that, and then settle down and then continue to do whatever it does. But some of them are completely attached. Now corals, yeah, here we go. So this is the attachment part, and then the body with the gastrovascular cavity inside and a bunch of tentacles. So here we're looking at the live one. So Medusa versus a polyp, right? Now we're looking at something which is known as a mushroom coral. It's a single one. It's not a colony. It's a single coral which creating the structure which looks like this. They start small and then they're growing much larger than that. And that one is about yay big, so about a foot in length. And then here we're looking at the colony of the corals, and those ones known as the honeycomb corals because, well, it looks like a honey structures. But as a matter of fact, this one was where the single coral polyp was residing. So they are small, but there is a bunch of them, so it's a colonial coral. And yeah, that's a side view. That one's something which is known as a horn coral. Horn coral because on that particular side, whenever you're looking at, I don't think I have a horn coral, but that's not a horn coral, but basically you're looking on one side, it looks like this, right? And then whenever you are flipping it over, it starts looking like a coral. That's not a coral, but it's similarly looking like this. So it looks like a horn, I guess. So that's actually a part of the endoskeleton for the mollusk. We'll talk about that later on. So what is the body form of the organism? That's a polyp. Now here we have some more corals. This is the coral I brought. That's called the brain coral because I guess it looks like a brain. So a typical coral, yeah, honeycomb. Yeah, see right there. Looks like a tiny little bit of horn, so horn corals. Now we're looking at the abelia. Abelia is the polyp, but it will have two different, very distinct polyps. It's a colonial polyp. Those ones with the tentacles are feeding polyps. Tentacles, they will be able to grab the food and digest it and then spread the wealth around, so to speak. Those, and usually they are at the tips. They're feeding, so once again, they are trying to increase the collecting area for whatever food they are collecting. And now there is a different types of the polyps, which are right here. Those ones are known as the reproductive polyps. You can see that they are literally creating a tiny little bitty jellyfish inside. So that jellyfish will literally pop out and it will be a tiny little bitty one. And it will start free swimming, and then there will be another one popped out and so on. Why? Sexual reproduction. Because the polyp by itself, if it's one of them right there, it's attached to the substrate, another is right there, it's kind of hard for them to exchange any genes. Free swimming medusas, however, can exchange the genes, and then we'll end up with a larvae, and larvae will grow into another colony. Make sense? So make sure you will be able to identify which one is feeding polyp, and then which one is the medusa of reproductive polyp. Reproductive producing medusas and the feeding containing the tentacles. And here we have a question saying, why does a feeding polyp need tentacles? To capture the prey and then get inside and digest it. Why does a reproductive polyp contain the medusa buds? Medusas are free floaters. easier to exchange the genetic material. Now here we're looking at the table. So here's the picture showing you the feeding with the polyps containing medusas, and you can actually see them under higher magnification. So here we go. Jellyfish, is it a polyp? No, generally a medusa, right? No colonies, it's a single form. Have a two body forms. Generally, most of the jellyfish lacking the polyp. They might have, but it's extremely short, so we're not going there. Most of the jellyfish kind of skip the polyp phase all over. They're mobile. They're not a very fast swimmers, but they definitely are swimmers. And sessile means sitting down, so no. Then C, anemone, is it a polyp? Yes. Medusa, they do have a short, but we'll skip it for the simplicity's sake. Colony of the polyps, some of the sea anemones can be colonies. Generally, they are solitary creatures. So have both forms, not really. Mobile, again, in most of the cases, it sits on the bottom and just waving its tentacles. In occasion, it can start swimming, but not really. So it's generally considered to be a sessile organism. Coral polyp, does it have a medusa? Sometimes for the reproduction they do, but we're not saying it has. Now colonial, can be colony polyps, can be a singular one. And then not a mobile, definitely sessile. Obelia, which is a type of the polyp as well as the medusa, right here. We have both of those, and it's a colony, and it has both of the forms, and some of them are sessile, some of them mobile. And then Hydra is the polyp, does not really have a medusa form, not a colony, and it's somewhat mobile, again, can do that particular Hydra seesaw, but generally sitting and waving its tentacles, so considered to be a sessile. And then the next group, known as the Ctenophora or comb jellies. They are relatively small creatures, and one of the very important characteristics that they do not have a cnidocytes. Cnidocytes, they are lacking of those stinging cells. See. They do have a radial symmetry, sometimes referred to as biradial symmetry. So, but in this particular case, what's really important, they have pretty much the same structure. A sac within the sac, tentacles, but the tentacles will be lined up with the sticky stuff. So this is how they grab the food. And they are free swimmers. They are swimming around. Whatever plankton or zooplankton gets in will be digested. But now, those guys have an interesting structure, the structure known as the traveling anus. Remember, the medusa, for example, the jellyfish, will get whatever, let's say, fish gets in, whatever is not digested will be throwing up. The research, and there was a paper published not so long ago, saying that whenever the ctenophores will have the gastrovascular cavity filled up, they generally do not throw up, but somewhere randomly, so far, we don't know what exactly triggers it, but there will be an opening on the side of the comb jelly, and all of the waste will be expelled that way. So it's not coming out from the mouth; it will be a random opening, everything expelled, the opening closes down, and it's continued to feed. And then the next time when it needs to expel the waste, the opening will be created somewhere else. So traveling anus, interesting, right? I think it is kind of neat. So Postan, it's a tiny little creature. So yes, looks like a jellyfish, hence the name comb jelly, but we figured out they initially don't have the cnidocytes, and then we are separate them into a very different phylum. So name the hollow cavity inside of the gastrula. And let's see if it has, but I don't remember if we were talking about. Basically, the cavity, yeah, here we go. Hollow cavity known as the archeron, right? And I think, yeah, hollow cavity inside of gastrula. This is what will turn into the digestive system later on. Name the opening of the archeron to go outside. That would be a blastopore. Name 3 germ layers, ecto and the mesoderm. Which animals have earliest form of nervous system? That would be the Cnidarians. Sponges do not have nervous system per se. They don't have specialized cells. So Cnidarians. Name the phyla studied in the lab have gastrovascular cavity. Cnidarians and Ctenophores. Which phylum of animals have the first complete gut? Essentially Ctenophores, because they have that particular traveling anus. What is the term used for animals that have 3 germ layers? Triploblastic. What is the special defensive feature Cnidarians have? Cnidocyte. Cytos means cell, right? Cytology is the science part of biology about cells. What is the distinct characteristics that set Ctenophores apart from Cnidarians? Absence of cnidocytes. And the reflection questions, you are generally doing an awesome job on that. I really enjoy reading what you learn, what you observe, and some of you actually putting like, well, we could add a little bit more of this or that. And if I can, I will change the lab a little bit in future generations of the students, of course. So please keep up a good job. And once again, at least a paragraph, 1 sentence is not a paragraph. So even if it's a super long sentence, split it into a 2 or 3, then it's for sure a paragraph, okay? Other than that, we are done for today. See, it was kind of easy. Next time we'll go into a nasty worms, and I'll show you some of the examples. But other than that, we are done for today. Let me stop the recording.